Now we are going to discuss electron transport chain and ATP synthesis. You have already studied about glycolysis and Krebs cycle. This electron transport chain means cellular respiration. So we will discuss now different stages of cellular respiration. There are overall 4 stages, that is glycolysis, PDH, means pyruvate dehydrogenase complex, Krebs cycle, and electron transport system. Whenever glucose molecule is taken by our body as a food, as a part of food, it enters into glycolysis, where this glucose molecule is converted into pyruvate. After conversion into pyruvate, it is converted by pyruvate dehydrogenase complex into acetyl-CoA, which enters Krebs cycle. There it is converted into carbon dioxide and water, and lots of FADH2 and NADH is being produced. These are electron donors, and finally these entered into electron transport system. There they give electrons for the generation of ATP, which is source of energy ultimately. Okay. In eukaryotes, electron transport and oxidative phosphorylation takes place in inner membrane of mitochondria. These processes reoxidize NADH and FADH2, which has been formed from the citric acid cycle in mitochondrial matrix, glycolysis in the cytoplasm, and fatty acid oxidation in mitochondrial matrix, and trap the energy released as ATP. In oxidative phosphorylation, major source of ATP is being synthesized in the cell. In prokaryotes, components of electron transport and oxidative phosphorylation takes place in the plasma membrane. Now let us see about redox potential. You know, in case of oxidation, there is loss of electrons, and in reduction, there is gain of electrons. In chemical reaction, if one molecule is oxidized to another, then another must be reduced. That is, oxidation reduction reaction involves transfer of electrons simultaneously. When NADH is oxidized to NAD+, it naturally loses electrons. When molecular oxygen is reduced to water, it gains electrons. So here you can see in this reaction, there is oxidation of NADH and into NAD+, and there is combination of hydrogen ion with half molecule of oxygen to produce water. Oxidation reduction potential, E, or redox potential, is a measure of affinity of a substance for electrons and is measured relative to hydrogen. Positive redox potential is substance having higher affinity for electrons than hydrogen, so would accept electrons from hydrogen, for example, oxygen, a strong oxidizing agent. Negative redox potential, having substance has a lower affinity for electrons than does hydrogen, would donate electrons to H+ forming hydrogen, for example, NADH, a strong reducing agent. For biological systems, standard redox potential for a substance E0 is measured at pH 7 and expressed in volts. In oxidation reduction reaction, electron transfer is occurring as total voltage change of the reaction, that is change in electrical potential delta E, is the sum of voltage changes of individual oxidation reduction steps. Standard free energy change of a reaction at pH 7 known as delta G naught is calculated from the change in redox potential delta E naught of substrates and products. In the reaction, you can see delta G naught is equal to minus NF delta E naught, where N is number of electrons transferred. Delta E naught in volts, V. Delta G naught is measured in kilocalories per mole, and F is Faraday constant. A reaction with a positive delta E naught has a negative delta G naught. That is, reaction is exergonic. Thus, for the reaction, here you can see in the reaction where NADH is oxidized to NAD+, there is delta E naught equal to 1.14 volt, whereas delta G naught is minus 52.6 kilocalorie per mole. Means where there is delta G naught in negative form, this reaction is exothermic. Means lot of energy is being produced as a result of this reaction. That is oxidation of NADH into NAD+. Electron transport from NADH. Comparing the energetic of the oxidation of NADH. As you have already seen, in case of oxidation of NADH into NAD+, there is delta G naught equal to minus 52.6 kilocalorie per mole. And synthesis of ATP from ADP and inorganic phosphate into ATP, in that reaction, delta G naught is plus 7.3 kilocalorie per mole. That is endothermic reaction. Means in the formation of ATP, energy is required for different steps. Thus, the oxidation of NADH releases sufficient energy to drive the synthesis of several molecules of ATP. NADH oxidation and ATP synthesis do not occur in a single step. Electrons are not transferred from NADH to oxygen directly. Rather, electrons are transferred from NADH to oxygen along a chain of electron carriers called the electron transport chain, also known as respiratory chain. Here you can see organization of ETC complexes. Electron transport chain consists of three large protein complexes embedded in the inner mitochondrial membrane as shown in this diagram. NADH dehydrogenase complex is first complex, that is also known as complex 1. Next comes succinate Q reductase. Third one is the cytochrome BC1 complex, that is complex C, and fourth is cytochrome oxidase, that is complex 4. Electrons flow from NADH to oxygen through these three complexes as shown in this figure. Each complex contains several electron carriers that work sequentially to carry electrons down the chain. Two free electron carriers are also needed to link these large complexes. That is, first one is ubiquinone, also called as coenzyme Q, and second is cytochrome C. So we have already discussed all these protein complexes as well as electron carriers involved in the electron transport chain. Now, ATP synthesis or oxidative phosphorylation. NADH and FADH2 are oxidized by electron transport through the respiratory chain for synthesis of ATP. Energy liberated by electron transport is used to create a proton gradient across the mitochondrial inner membrane that is used to drive ATP synthesis, which is chemiosmotic hypothesis in presence of ATP synthase enzyme. Thus, the proton gradient couples electron transport and ATP synthesis, which is not a chemical intermediate as in substrate level phosphorylation. And that ATP synthase enzyme was originally known as ATPase because without input of energy from electron transport, the reaction can reverse and actually hydrolyze ATP. In short, you can remember, better for revision, electron transport down the respiratory chain from NADH oxidation causes hydrogen ions to be pumped out into the intermembrane space by three hydrogen ion pumps, that is NADH dehydrogenase, cytochrome BC1 complex, and cytochrome oxidase. Free energy change in transporting an electrically charged ion across a membrane leads to the formation of electrochemical proton gradient. The pumping out of the hydrogen ion generates a higher concentration of H plus ions in the intermembrane space and an electrical potential. So, the side of the inner mitochondrial membrane facing the intermembrane space being positive. Protons flow back into mitochondrial matrix according to electrochemical gradient through the ATP synthase, which drives ATP synthesis. The ATP synthase is driven by proton motive force, which is the sum of the pH gradient and membrane potential. FADH2 is reoxidized via ubiquinone. Its oxidation causes hydrogen ions to be pumped out only by the cytochrome BC1 complex and cytochrome oxidase. So the amount of ATP made from FADH2 is less than from NADH. Measurements show that 2.5 ATP or 3 molecules of ATP are synthesized per NADH oxidized, whereas 1.5 ATPs are synthesized per FADH2 oxidized. Now let us see summary of electron flow by this diagram. Process of electron transport in mitochondrial membrane. In mitochondrial inner membrane, we have four different complexes, as we have already discussed that four different complex of protein which are embedded and involved with the task of electron transport and ultimately converting it to the water at the end by attaching with oxygen molecule, as uh, you are looking at in this diagram. While oxidizing NADH and FADH2 to generate substantial amount of energy and ATP molecule, now in this case we will see only the role of three complexes which are present in inner membrane of the mitochondria. Complex 1 transports electrons from NADH to coenzyme Q. It contains more than 40 protein subunits and abound FMN, flavin mononucleotide site, numerous iron sulfur centers. It translocates two protons in intermembrane space and per NADH oxidized, so four protons for a single NADH. Now complex 3 accepts electrons from reduced coenzyme Q and transports them to cytochrome C. It contains 11 protein subunits and 3 cytochrome regions, 1 iron sulfur center. It translocates 4 protons per 2 electrons are transferred to cytochrome C. Each cytochrome C accepts 1 electron, and so for transferring 2 electrons, we need 2 cytochrome C in this place. Now the last complex 4. Cytochrome C oxidase accepts electron from reduced cytochrome C and transports them to the molecular oxygen for reduction it into the water. It contains 13 protein subunits, 4 cytochromes, and 1 copper site. It translocates 2 protons per 2 electrons and also consumes 2 protons from matrix in generating 1 molecule of water from half molecule of oxygen. Now, let us discuss about coupling of electron transport and ATP synthesis, because final result of this cellular respiration is synthesis of ATP for energy generation. This is divided into 2 steps. First one is electron transport across mitochondrial membrane, which will help to generate proton gradient across inner membrane of mitochondria. And second step is rotation of rotor unit of ATP synthase enzyme to produce ATP by combining ADP with inorganic phosphate. There is a link known as coupling of electron transport chain and ATP synthesis. Electron transport occurs along the inner mitochondrial membrane. Electron transport complex 1, 3, and 4 translocates proton to intermembrane space, which helps to generate a proton electrochemical gradient negative in the matrix by building high concentration of protons in intermembrane space, contributes to proton electrochemical gradient that drive ATP synthesis by complex 5 in the presence of ATP synthase. And thus protons are returned to matrix and ADP combines with inorganic phosphate to make ATP. And finally, as a result of this cellular respiration, ATP is being synthesized. Thank you. Thank you very much.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.